Hello, and welcome to the relationship between HIV/AIDS and the human immune system. So to start us off, what is HIV/AIDS? Let's begin with understanding what HIV is. HIV stands for Human Immunodeficiency Virus. According to HIV.org, HIV is a virus that attacks immune cells. This leads to the infected person being more susceptible to other infections and viruses. HIV is spread through contact with certain bodily fluids of a person who is infected with HIV. However, if HIV is left untreated, it ends up leading to AIDS. So now, what is AIDS? AIDS stands for Acquired Immunodeficiency Syndrome, and according to HIV.gov, AIDS is a stage of HIV. During this stage, the body's immune system is now extremely damaged due to HIV. The qualities of a person's immune system when they get to the AIDS stage are when a person's CD4 cell count falls below 200 cells per cubic milliliter of blood. To better understand this, a healthy human has a CD4 cell count between 500 and 1600 cells per cubic milliliter of blood. When a person develops one or more opportunistic infections, regardless of their CD4 cell count, they also have AIDS. Now that we know a little bit about HIV and AIDS, let's take a look at the journey HIV takes through the human immune system. Let's begin by understanding the human immune system's chemical and physical barriers. In the case of the HIV virus, the most common way of contraction is through unprotected sex. According to the National Institutes of Health, the human immune system has chemical and physical barriers like mucus that lines the mucosal membranes, antimicrobial peptides found in bodily fluids, stomach acid, and a health microbiome. All of these different aspects come together to form the first line of defense to prevent the virus from entering the body and initiating an infection. Sadly, HIV still gets past these physical and chemical barriers. When the HIV virus gets past the physical and chemical barriers of the immune system, it establishes a local infection site. Here, at the local infection site, you can find the innate immune system in the infection. In our case, that's the HIV virus. This is where the battle between the human immune system and the HIV virus begins. The innate immune system consists of different types of cells. The first cell we are going to look at are called tissue-resident macrophages. These cells are found everywhere in your body, from your liver to your brain. Macrophages recognize the HIV virus by identifying damage-associated molecular patterns or pathogen-associated molecular patterns using pattern recognition receptors. Different antigens have different DAMPs and PAMPs. According to the National Institute of Health, the DAMPs that come from the HIV virus are high-mobility group box 1, mitochondrial DNA, and certain intracellular nucleic acids. The PRRs found on the residential macrophages that detect these PAMPs are toll-like receptors and receptors for advanced glycation end products. Normally, macrophages would send signals to recruit other innate immune cells, but things do not go as planned with the HIV virus. Macrophages still send signals, but according to PubMed, HIV infects macrophages and replicates, meaning that the first line of defense of the human immune system is one of the main ways that the HIV virus replicates. Macrophages move around containing pieces of the HIV virons, spreading the virus to other cells. Another innate immune cell found in the local infection site are dendritic cells. Dendritic cells engulf pieces of the HIV virus. Just like macrophages, the HIV virus exploits the job of the dendritic cell of transferring antigen. Just trying to do their job, dendritic cells are also helping to spread the HIV virus. Now that we have learned how HIV affects the innate immune system, let's look at how the HIV virus affects the adaptive immune system. We left off at dendritic cells engulfing antigen from the HIV virus and transporting it through the lymphatic system to reach T cells. HIV virus specifically binds to class II CD4 plus T cells. As a CD4 plus T cell and HIV antigen bind, the HIV virus injects its genetic material into the CD4 plus T cell. Once inside of the T cell, the HIV virus replicates. According to PubMed, once inside of the CD4 plus T cells, the HIV virus kills the T cell through a process called pyroptosis. This process is when a cell's membrane ruptures, killing the cell and releasing the cell's contents. And the contents released by the T cell are inflammatory cytokines that, when in contact with other T cells, trigger pyroptosis. This creates a dangerous cycle that leads to the immune system collapsing. We've seen the detrimental effect HIV has on T cells, specifically CD4 plus T cells. Now let's look at what kind of an effect HIV has on B cells. While the primary target of HIV is CD4 plus T cells, B cells are indirectly affected due to their reliance on helper T cells for proper activation and function. Typically, in the presence of viral infections, naive B cells respond to an antigen by migrating to T cell-rich areas of lymphoid tissues, becoming activated, and initiating a germinal center reaction that selects for B cells with improved antigen binding. Finally, the B cell exits the germinal center as either a memory B cell or plasma cells according to PubMed. While the primary target of HIV is CD4 plus T cells, B cells are indirectly affected due to their reliance on helper T cells for Proctor activation and function. Additionally, according to the National Institute of Health, HIV causes polyclonal activation, a nonspecific immune response. Polyclonal activation occurs when a high concentration of an antigen causes most B cells to proliferate and differentiate regardless of their antigen specificity. The chronic immune activation induced by HIV also gives rise to increased production of antibodies, elevated levels of immunoglobulin in the blood, and exhaustion of B cell resources over time due to persistent activation. There is also a real risk of B cell malignancies. HIV-associated chronic immune activation predisposes individuals to B-cell-related cancers, such as non-Hodgkin lymphoma and Burkitt lymphoma, as noted in Frontiers in Cellular and Infection Microbiology. HIV's impact on B cells, T cells, dendritic cells, and immune cells illustrates the virus's ability to broadly disrupt the entire immune system, making the disease a lifelong struggle for those who have it. While there is no cure for HIV, there are treatments that provide those infected with some hope. According to UNAIDS, antiretroviral medicines are used to treat HIV, but not cure it. They prevent the process that replicates the genetic material of HIV, inhibit the reverse transcriptase enzyme, and prevent the breakdown of proteins to slow production of new virus particles. There are various different kinds of antiretroviral therapies that can be used to treat HIV, such as nucleoside reverse transcriptase inhibitors, non-nucleoside reverse transcriptase inhibitors, and protease inhibitors. These medicines are often used together, known as highly active antiretroviral therapy. The combination is very effective at suppressing the virus because it targets multiple locations of the replication cycle, according to the National Institute of Health. HIV does wreak havoc on an individual's immune system, allowing for opportunistic pathogens to come invade. But with the proper treatments, those infected can live long, healthy lives. Thank you.